Hi, everybody. Thank you for joining. Just a reminder, we are going to start at 6:05, so just in a few minutes. During the talk, please keep your videos off and your microphones muted, and we will be starting shortly.
Kia ora, everybody, and welcome to tonight's Blake Ambassador Speaker Night 2021. We are very happy that you have joined us in person and via Zoom. We have a very exciting night planned for you. We have three great speakers here with us. One will be joining us on Zoom as well. Every speaker will have around 25 minutes to present, and we would like you to, if you are via Zoom, to post your questions on the chat box. But if you're here in person, to hold your questions until the end of the presentation of each of the speakers. If you are joining us via Zoom, please remember to keep your microphones off as well as your cameras. And for everyone that's here tonight, we will have some pizza after, and you can talk to the speakers in person. Before I introduce everybody that's joining us tonight in this talks, I'm going to tell you a little bit about the Ambassador Program of Blake. So every year, young New Zealanders aged between 18 and 25 joined the program, the Blake Ambassador Program, which it's in partnership with NIWA, the Department of Conservation, and GNS Science to provide young adults with a unique opportunity to have a hands-on experience and do science and get involved in projects that are very important to the environment. Today, we're excited to present three speakers who have recently completed their Blake Ambassadorships this summer, and we will hear from all of them tonight. They did this ambassadorships during the summer, and today we're going to first welcome Amy Burney. She was the Blake NIWA Ambassador for Climate and Atmosphere. She completed her Bachelor of Science, majoring in chemistry at the University of Canterbury in the Physical and Chemical Science Department. She has an interest in the boundary between chemistry and physics, and she loves the outdoors. She's named rock climbing, hiking, and canyoning, and she also like to do creative activities as well. In her ambassadorship, she learned about gas measurements and monitoring at NIWA, and she's going to speak to us tonight. Welcome, Amy, to our talk tonight.
Hi, guys. Obviously, I'm not here in person, but I'll just sort out sharing my screen with you. Right. Can people see that okay? Can you guys see the screen okay there?
Yep, you're all good to go.
Cool. So I haven't really planned what I'm going to say tonight, but I'll just go through basically what I got up to when I was doing my ambassadorship. I spent the first 2 weeks of my ambassadorship up in Wellington. I'm from Christchurch originally. Up in Wellington at the Greater Point site, where they mostly do research into atmosphere, which is what I was looking at, and also quite a bit into sea ice and things like that. They mostly do data crunching there, so it was a lot of sitting at a computer for me, but still a good time. And we also got out to their Baring Head site, which is the picture you can see on the screen at the moment. So on you'll be able to see on here in the top right, I went around a few of their various labs that they have on site there, and they the main thing that they investigate is the concentrations of carbon dioxide and methane gases in the atmosphere, and also measure the radiocarbon levels of each to determine the source of the carbon dioxide. So on the top right of your presentation, you'll see how they process the to determine the concentration of carbon-14 to carbon-12 as fossil fuels are severely depleted in carbon-14. So you can calculate a fossil fraction of where the carbon dioxide comes from. What I found quite amazing is to process that they need, I think it was 100 liters of gas to determine this percentage of carbon-14. And if they want historic measurements, they have to take this from ice core samples from the Antarctic, which, as ice is only 10% air gap space, that's an awful lot of ice to process to get a measurement out. The bottles in the image on the screen, they were all of the testing bottles at the Baring Head site. So Baring Head is a very exposed site right on the bottom right corner of the North Island. So it gets perfect southerly winds in from the Antarctic. And they've been taking measurements there since of CO2 since 1972, which is the longest running CO2 measurement, second longest running CO2 measurement in the world. It was quite amazing to see the site and actually have a hand in checking all of the equipment and maintaining it. The site's mostly automated at the moment and has a continuous data stream to the main Wellington site. So they don't have to be there all the time, but continuous measurements are being made. They go out there once a week and maintain everything, check tank pressures and stuff like that. The main thing that the data there is used for, as well as seeing how CO2 levels are changing over time, is it's a good blank, as it only has, if you take a measurement in the strong southerly, it's only air coming from the sea, which if you compare it to other directions of wind, you can see where the CO2 is coming from, which areas of land are the main contributors, which is what one of the people I met there is doing. They've got, their plan is to have a national scale model of where the air packets are moving. And they're quite interested in seeing how old growth versus new growth forest impacts things. It was thought that young growth forest is the best for trapping CO2. However, there's some idea that old growth around Fiordland may actually be a good carbon sink, but they're still looking into that. I also got a chance to test some of their equipment before they used it in the field. So the picture of me on here is of me testing for leaks on metal pipes, which are used to dry out air samples as they're being taken, so there's no moisture or water in the sample. The two graphs here, the bottom one is showing how they are comparing the carbon 13 and 14 measurements of methane to find the source of the methane. And the other graph is one that I actually made. They have data of methane from many different sites, and we were trying to see whether we could combine the data from these various sites into a larger scale model to give a better idea of the fossil fraction. So how much of the methane in the atmosphere or CO2 is from fossil fuels. In my second week in Wellington, I did some more of the data crunching. We didn't really form any conclusions. The data wasn't really comparable, but it was really good to have a chance to see actually where all of these measurements were going and what could be done with them. But what I did get to see, which might be of more interest to you guys, is the Invertebrate Collection, which was this huge room with like old library style rollout shelving with, you can see three things on the bottom here. If I'd been able to hear you, I would have asked you what the three things are. The three things on the bottom, so on the far left, we have a ram's horn squid, which I thought was really neat because they have a little shell inside their heads, which they use for buoyancy, which I've seen the shells on the beaches actually, but I never thought they would have come from the head of a squid like that. Then you have a giant isopod, which was pretty huge, as you can see there. And on the right, we have an Antarctic worm, which has scales and bristles and a giant jaw. And I don't think I'd want to meet that if I ever went swimming. You can also see here, I had a good chance to go and see the supercomputer at their Wellington site, which was quite an experience. They first took me to their old site of data storage, which was, I thought was big. It was a room with a whole heap of tapes, like a giant room. It looked like a big computer with stacks and stacks of old tapes and a robot arm pretty much that went out and picked the right tape and plugged it in if someone wanted to retrieve the data from it. But then we came to the actual supercomputer, which had a building to itself, and it was a building inside the building to protect it from anything that might happen. And they use it for not only NIWA itself, but also quite a few of the academic institutes around the country, which is quite cool. And yeah, it was massive. And to protect all the storage there, they actually had a separate room full of batteries, which you can see there, which is a lot of batteries, but apparently only lasts for 15 minutes worth of power. And it's just the backup system before the generator backup kicks in if the power goes out. And there was also a whole room of giant cylinders of fire gas, like fire putting out gas. It was just crazy to see how much has to go into just protecting this one room of data storage. And after I'd spent 2 weeks in Wellington, I went down to Lauder, which I imagine most of you don't have any clue where it is, unless maybe you've done the Otago rail cycle trail. But it's this tiny town in the middle of central Otago with nothing there. There was a pub when I was there, but no longer. But NIWA has a site there with 8 full-time staff, basically a group of nerds who like looking at the sky and measuring the gases there. I really loved it down there. I got a chance to see all of their different equipment, help out running most of it. It was pretty cool. Down there, they have, their main focus is on measuring the ozone in the atmosphere. Originally, they were set up to look in the 1960s to look at the ionosphere in the interest of satellite interference. But since the discovery of the ozone hole, the main focus has been ozone. And they have a huge variety of different ways of measuring the ozone. So that they take total column measurements by shining a laser, saying the top up into the sky quite often to measure how much ozone is between the ground level and the level where the satellites are. They also take lots of light meter readings, and in the top right here is a picture of their receiver. So it downloads the data from a satellite that travels over top and saves it. They also take the ozone measurements at particular altitudes using an ozone sonde, which it's basically a giant party helium balloon. The one in the picture here is actually a half-size one. We had erected a new radio receiver whilst I was there, and we were just testing out to see how well that worked at retrieving the location data from the ozone sonde. So that if that one looks big, the real one is huge. I helped them set up this ozone sonde, so it's a little cell that detects how much ozone is present and relays back via radio to the Lauder site as this balloon floats up into the atmosphere, which is quite cool. So you can see how it varies from ground level all the way up. And we were also tracking the location, which you can see. It goes a long way, normally out to sea, and it pops when it gets to a particular height. Yeah, so it was pretty much all I had to say about what I did, but it was really interesting hearing all about the CO2 and methane measurements up in Wellington and the ozone research and down in Lauder. Yeah. Does anyone have any questions?
Thank you, Amy, for that. We're going to give a few minutes for people if they want to write in their chat if you're in the Zoom meeting. So please pop your questions in the chat. I'll be reading them out here. I'm going to ask in the room if we have any questions for Amy. Any questions? I have one question. I'm going to quickly ask you about why is it important to know where the carbon dioxide comes from?
Well, it's just good to know, like if we know where it comes from, we know how we can tackle the causes of increased carbon dioxide and especially methane, rather than just assuming where the sources are. We can actually try and work it out and approach the people who are causing the issues, I guess.
Thank you. I have another question here. What is the ionosphere? If you can tell us a little bit about it.
I'm not, that's more of a physics question, I guess. The ionosphere is one of the levels of the atmosphere, which is, I think it's the upper level. I would actually have to Google it, to be honest. But it's not where the chemistry that we're interested in is happening. It's higher up than that. We're more looking at tropospheric chemistry when we're talking about ozone, or potentially stratospheric, which is the next one up from the troposphere.
Okay, there's another question. It says, why do you not want water in your samples?
So the way that they measure the samples, it's determining like what chemicals are in there, or what gases. So water can actually interfere with the oxygen signal, I believe. So it just makes a less clear measurement result, I guess.
Okay. I have another question from the chat group. It says, great to hear about your experience, Amy. Has your experience influenced your future plans, or how do you see yourself contributing to New Zealand in the future?
I think it has influenced my plans. I've always been interested in both physics and chemistry, which I guess atmosphere is the perfect kind of bridge between those for me, especially as I'm interested in the environment. But having the opportunity to see what NIWA does kind of gave me an idea of how that all fits together, how the chemistry of the atmosphere is kind of vital for seeing how we're impacting the environment. Yeah.
Amy, and from all the things you learned during this ambassadorship, what would be your advice for other people that would like to apply to be a Blake ambassador?
I say go for it. I was quite unsure about applying myself. I didn't think I stood a chance, and I didn't know whether it would be for me, but I went along and it was like a great opportunity, great fun. I learned a lot. I was basically taking notes the whole time, not because anyone asked me to. There was no expectation of what I was to do really, but it was just, I wanted to learn everything they had to tell me. It was great fun. Yeah.
Great. Is there anything you would like to share with the people here or at Zoom that you would like to say as a final message before we continue with our next speakers?
Not really, just enjoy what the others have to say. I'm sure they had a great opportunity and enjoyed their time too. Thank you for listening.
Thank you. Can we give Amy a round of applause? Thank you very much. We're going to continue with our next speaker. The person that's coming following next is Janie Ann Swetman. Can you guys still hear me and see me? Yeah. Janie Ann is a Blake NIWA ambassador in aquaculture. She is a master's student at Massey University, and she is researching the long spine sea urchin in northeast New Zealand and Rangitahua. She loves the outdoors and the ocean, and she does things like tramping, surfing, and snorkeling. She's a very creative person too. She loves and enjoys art, and in particular painting the creatures of the ocean, which I'm sure she can tell us a little bit about it in her presentation. Jenny spent 2 weeks at the NIWA Northland Marine Research Center in Bream Bay, and she learned about aquaculture, and I'm gonna open the floor for her so she can tell us about her experience.
Share screen. Okay. Share screen first. Sorry guys, we're just sorting out Zoom.
This is you?
Yeah.
But it's that one. Yeah.
Okay,
Cool.
can everyone see that? Why is it not going?
Okay, give us some thumbs
Oh, yeah.
up
Okay, cool.
in your chat if you can see the screen.
No.
Okay, thanks,
Now it
Amy.
should be working. Okay. Hi, everyone. I'm Janie Ann, and yeah, I did the NIWA ambassadorship. So yeah, starting with a bit about me, as Carla said, I'm doing a master's at Massey University on the long-spine sea urchin, looking at population genetics and size structure to understand their history and demography in New Zealand. Yeah, they have quite a potential to impact our fisheries and biodiversity from urchin barrens, so that's kind of why I'm interested in them. Another thing about me is that I really love the ocean, and I really wanted to make sure that we have a healthy ocean that can be there and healthy for future generations. And I'm interested in addressing climate change, pollution, and fisheries, because I think that they're the things that are really important. Yeah, so I was really interested in this Blake ambassadorship because I wanted to learn about aquaculture and yeah, just how it can be used in terms of having a sustainable fishery and how we can provide food for people, having a less of an impact on the ocean. So I went to the NIWA Northland Marine Research Center. It's New Zealand's largest land-based aquaculture facility, and it's up in Ruakaha, just by the beach, because it gets all the water from the beach. And yeah, so it's a research facility, and they have lots of research projects, but they commercially sell their. So they work with, quite a few species, as I said, catfish and seaweed, which I'll mention a bit later on. And then rāpoka and kingfish, they sell commercially, and then they also work with salmon and snapper. So I'm going to talk about the kingfish because that's what they were doing when I was there, and that's what, I mainly worked on. So yeah, kingfish, they can grow pretty big, and what's really cool at the NIWA is that you get to see the whole process. So from when they're eggs all the way to when they get harvested. So yeah, starting from where we get the eggs, the brood stock, they breed about 4 times a year, and this can be manipulated using the water temperature, which is another advantage of having a land-based marine facility because you can change the water temperature. And the eggs are collected in these nets when they're spawning, which is the water going through the tank. And then they go into here, and then the dead ones will sink to the bottom, and then we just want these eggs, and then they can go to the hatchery. And something I found really interesting is that they use genetics to work out which fish are breeding with each other, and if say there's one fish that's not really breeding, don't really want that, or one fish that's breeding lots, and then they can switch around the fish to get what they want. So in the hatchery, pretty much you look at the eggs. They look at the eggs under the microscope, and you can see what stage they're at. So here they're earlier on, but then you can see these ones on the left. You can see the embryos are formed. So that's when they're later on in the stage. And yeah, also want to know if there's lots of deformities, then that's not going to be a very good batch, and need to make sure they're fertilized so we actually get fish. And after this, we get the larvae, and then the larvae go into the nursery. And yeah, they grow really fast. So when I was there, I could see the difference day by day. And when I went away for the weekend and came back, then they were suddenly bigger. So they're really small, but yeah, you can see from here to here, they start growing, and you can see their eyes, and they change color a bit. And yeah, they get fed rotifers and artemia, kind of changing on how big they are. And then after this, they get sorted into size. So they'll go into size of their tanks. And just kind of the size is just kind of to make sure that they're all similar size. So with feeding and stuff, they'll have a similar chance, I think. So to sort them by size, they went through this blue container here, and the small ones will just go through, and the bigger ones will stay there. So these are the fingerlings. The next fish, the ones I was working with, were 5 to 7 weeks old. These were my favorite because they're so small and cute. So yeah, I spent quite a lot of time in the nursery, and just every day you have to do the feeding and cleaning the tanks, and then the oxygen and temperature of the tanks needed to be checked for all the tanks. And yeah, so, and there were some other tasks, like for example, we check the weights of the fish, so get a whole heap, and then, get like a bucket of fish, and then you count them out and work out what the weights are. And then we do more fish weights when they're bigger, kind of gets done along the process, just to assess their condition. So in these photos, you can see, get them out of the tank with the net, and this is definitely the job where I got the most completely soaked, because they were just going everywhere, and they're actually quite hard to catch. Like, you wouldn't think that they'd be so hard to catch. And then they go in here where there's aqueous, which is just like, it just like slows them down and sedates them a bit so that they're not so stressed. And then they get weighed and measured, and then they just go back into the tank. Yeah, so moving the fish between the different areas was quite a process, because obviously when they're small, when they're like the larvae, quite easy to move because they're tiny, but they get massive. So as they get bigger, it gets more of a process. So this stage, I think this was on my first day, and we were just putting the fish, and they were going down this pipe into the this container, and then yeah, we were just counting the fish, because we had to know how many fish there were. And then this got taken by a forklift to the next area, and then these ones here, these were actually broodstock being moved, and they're quite big, right? So it was quite a challenge to get these out of the tank and carry them. Yeah, and all the fish need feeding. And something that NIWA does is they do run feed trials. So looking at, for a company, if they have different feeds, looking at the performance between them. And yeah, I just find the feed really interesting, because lots of feeds have fish meal or fish oil in them, which it can come from an unsustainable source. So yeah, I guess ideally, if you're having this aquaculture facility, it'd be good if the stuff that's going into it is also sustainable. But yeah, interestingly, there is lots of things happening in that area. So using things like soy or insects and stuff, and I'm sure lots of other stuff in their feed. Yeah, and then finally they get harvested, and then they go off to restaurants. And I'll just talk about a few other things that were going on while I was there. So catfish, these fish that look kind of weird, are an invasive freshwater fish, and they prey on our native freshwater fish and eggs, and also our native crayfish. And they're really hard to kill, and they can survive really well. They can survive out of water for a while. They can survive in really low oxygen, and yeah, they just seem to be able to survive anything. So quite a challenge. But the goal here is that they're doing is trying to create sterile males, and then they'll breed with the females, and then those eggs won't be fertilized. So that will help control the population. But this project was very much in the start when I was there. So we were just trying to get them to have eggs. They didn't seem to like to breed. They didn't seem to want to have eggs. We had all different types of things to see what they might like these rocks or pipes and stuff. But then they did end up having eggs. Here's the eggs. It was quite exciting. We just checked them just before we were about to leave for the day, and then they had eggs. So it was all, we need to do something with the eggs. This is another part I found quite interesting. So they have a recirculating aquaculture system, which means that pretty much the water goes in from the sea, and then it's used with the oxygen and the feed with the fish. And then after that, instead of just going straight out to sea, it goes through a series of filters, which clean it and take all the nutrients out, and like, yeah, and then it can be used again. And I think like with different systems, a different amount of water will be reused, but you can end up using most of the water, which is really great in terms of, yeah, you're using less water. And also things like, it can be more environmentally friendly, because you don't have to, if you need to change the temperature of that water, then if it's going through, you're going to have less seawater coming in, so you'll have to do that less, which uses less energy. Yeah, the other really cool thing was the seaweed. So yeah, this is a picture of the seaweed, and they're collecting and growing them and looking at the life stages and stuff. And it's because, yeah, this seaweed, the red macroalgae, can be used to feed cows and livestock, and it can reduce the methane emissions they produce, which is pretty cool in terms of agriculture being a massive producer of greenhouse gases. So overall, I think, yeah, I really enjoyed meeting everyone and finding out the different paths that people took to get to their job. And I thought the catfish research was really cool because it can be used in a sort of conservation way, and the RAS system. And yeah, I really liked working in the nursery with the tiny fish, which you'll probably be able to tell from how many pictures I had of them. Yeah, and hopefully I'll be able to use the knowledge I got to help continue with my plans to help look after our moana. Yeah, and I guess, yeah, if anyone is thinking of being a Blake ambassador, what I would say is think about like what your strengths are and what your passions are and how they could relate to it. And also just like give it a go, but if you don't get it, I didn't get it, I applied, and then just give it another try, and then you might get it the next time. That's what I did. Thank you for listening.
Do we have any questions from the audience? Yes, Imogen, I can repeat for everyone listening on Zoom.
Was there anything that particularly surprised you working at the aquaculture facility?
So I'll just repeat, was there anything that surprised you working at the aquaculture facility?
I'm trying to think. I guess I completely just like going into it, I didn't actually know that much about aquaculture, so I wasn't really like sure what to expect. But maybe just like how many like eggs and stuff there are, like, and how many like they have to keep cutting it down each. Like they won't keep all the larvae, you know, there'll be tons of larvae, and then they'll only keep like a little portion of them for the next stage, and like it's like so like keeps decreasing by stage. Yeah.
I also have a question. So we kind of learn about aquaculture a little bit, and then it's different to actually see things happening, right? So was the feeding a big part of it? Like, was it a lot of food? How's the feeding? I don't know. Did you see much of that? Was it like quite shocking how much food you needed to provide for fish or things like that?
I don't think it was like that surprising. Yeah, so I did quite a lot of feeding because it was a good job to get us to do. But I don't think like, yeah, so I mean that for like a big tank of fish, it was like a thing of, it didn't seem like that much food. And like sometimes like so when I was there, apparently this has never happened, but like something with the pipe and things went wrong and the water couldn't, so then they just like didn't feed them for a few days, and apparently they're fine, like they're all good. Like except the little ones, they still got food, so they seem pretty resilient.
Okay, cool. I have a couple questions from the chat. The first one says, thank you for the interesting presentation. Your work is amazing. What an exciting life. My question is, why were you breeding the kingfish and were they endangered?
So it's just for, breeding them, I guess, like as a commercial product. So to sell them for restaurants and stuff. They're not endangered. Yeah, so that's the reason behind it. And I guess, yeah, so just as a commercial product to sell the kingfish. And I think sort of, I guess because NIWA is a research facility, they're sort of interested in how it can be done in the most efficient ways, sort of.
And this ties to this other question that was here. It says, did you get to eat any of the commercially bought fishes?
I did actually get to eat some fish.
Cool. I have another question from the chat. It says, do you know if the fish get given any antibiotics or medication, just such as to reduce lice infections? I believe that this is one of the criticisms against aquaculture. Also, do you know how many fish they produce per annum?
So I'm not sure about how many fish they produce. And I'm also not sure about the other question, but like, as far as I know, could be wrong. I don't think so. I guess they're pretty careful with sorting them with like anything that looks like it could have any sort of like infection or anything, then they won't keep those ones to try stop so that they won't spread to the rest of them.
We have a question here in the audience. Okay, so I'm going to repeat the question for the people on Zoom. So what were the pathways that people took to get into this research? Yeah?
Yeah, so lots of different things. I mean, yeah, there are a few people who had done, you can do like a 6-week internship there. So there were a few people who had done that and that had led on to, and then maybe some of them had like gone overseas and worked in some other aquaculture and then come back and worked there. And then, but there were also some people, like there was one person who, he must have been helping out, but he had just pretty much left school in the lockdown and started working there. He went straight into it. I guess it depends which kind of jobs you're going into at the facility. But there just seemed to be lots of people doing all different stuff, like someone who was, yeah, I guess it surprised me how like wider breadth of people. But yeah, I guess if you want to be doing the actual research, you do need to go the science path. But yeah, not if you want to be working on some of the other stuff, because there's lots of people just like, I guess, fixing problems all the time. So like just building stuff to fix the problem and like, yeah.
Awesome. There's a couple more questions in the chat. One says, how did you come across this opportunity? It sounds amazing.
So like, I don't know, I just saw it a few years ago. And yeah, so I think my friend did it. Yeah. And then as I said, I think the first year I saw it, I just missed it. And the next year I applied and didn't get it. And then, yeah, I applied and did get it.
Third time is a charm. One more question on the chat. It says, do you know if the energy consumption to run a fishery such as this is less, especially in terms of carbon emissions, than a traditional offshore fishing vessel?
I don't know. And yeah, that's something I'd be really interested in. Yeah, because I think there's, different sides to the aquaculture and non-aquaculture. And yeah, it'd be really interesting to know the comparisons.
Cool. And to finish off with questions, if nobody else has any questions here, did you do any exploring in your free time?
I went up Mount Mania and went to Rāpoka Beach quite a few times because it's right there. Yeah.
Awesome. Can we thank Janie Ann with a round of applause? Sorry. Thank you. Just wanted to make sure I said Ann. Very important. Okay, awesome. We are going to invite our third speaker. I'm going to escape this sharing now. So for the people that just joining us, you're welcome to ask questions on the chat, and we will be now presenting our third speaker. And this is Wednesday Davis. She was the Blake Dock Ambassador for hoiho and rāpoka from the University of Auckland. She's recently completed her PG dip in marine science, and now she's working towards her master's in marine science, researching behavioral interactions between cetaceans, birds, and their prey. She's originally from Mount Maunganui. She's also a diver. She loves fish and all the creatures in the ocean. She's worked as an assistant aquarist at the aquarium, and she recently completed also an internship with the Auckland Council. Wednesday spent 3 weeks in Dunedin and the Caitlin's this summer, and she worked with rangers and scientists about surveilling programs for the hoiho, the yellow-eyed penguin, and the rāpoka or pakake, which is the New Zealand sea lion. Let me welcome Wednesday Davis.
Hello, everybody. Sorry, I'm just getting my screen up. Oh, maybe not. Here we go. Awesome. So hi, my name is Wednesday. I was the 2020/21 Dock and Yellow-Eyed Penguin Trust Ambassador. So I did this ambassadorship with Giovanni Fobbs, who is a master's student from the University of Otago, and I'll be telling you guys a bit about our trip. So firstly, a little bit about me. Like Carla said, I'm a master's student here at Auckland University. So I study whales, dolphins, and seabirds and how they all work together in the ocean to help forage on food, which is pretty exciting. I'm using drones and the top of the line AI to hopefully automate it so I don't have to go through thousands of little photos from our videos. I also work at the aquarium. I'm a fish keeper. I'm a scuba diver and love exploring my big backyard. And I'm also a volunteer for Experiencing Marine Reserves, Aotearoa Lakes, and Sea Forward. So a bit about my ambassadorship. I spent 3 weeks in Dunedin and the Catlins. So I spent 2 weeks with the Dunedin dock team. So that included a bunch of really awesome rangers, as well as some summer ambassadors. I spent 1 day at Penguin Place, which is a rehabilitation centre, and 1 week in the Catlins with the Yellow-Eyed Penguin Trust. So just a little bit of background about the critters I worked with. So this is a New Zealand sea lion. This one here was hanging out on the beach. You can see it's just come out of the water and it's got some waters on its whiskers. So they used to be found all around the coastline of New Zealand, so all the way up to here in Auckland. But unfortunately, about 200 years ago, we started harvesting them. We started fishing them and wiped them out across the entire mainland of New Zealand, restricting them just to the subantarctic islands. They are still threatened by a range of different things, including overfishing of their food, which is squid and other big fishes, dog attacks when they're on land, and human disturbances and attacks. Luckily, they are slowly recovering and pups are now being born on mainland New Zealand. What do I mean by this? Well, meet Mum. So this is one of the pups from this year, so it's not Mum. But in 1993, a female sea lion called Mum had the first ever pup born in mainland New Zealand in over 100 years. Since then, more and more pups have been born here, including 11 of her pups. Unfortunately, she disappeared when she was about 24 years old, but her legacy lives on, and now there are more and more pups being born every year. And this year, there were over 15 pups born. So some of Doc's work, they monitor the haul-out site. So this is where the sea lions will come out to have a rest in between feeding, but also known areas of pupping. So a lot of the mums will return to the same beaches every single year to have their babies. They also microchip and tag the pups. They scan the microchips on adults to record the sightings, and they monitor the movement of pups as well. So even though the pups are born in the bush, the mums will actually move the babies from where they were born to little creches. It's kind of like a daycare system. Babies will hang out together with 1 or 2 sea lion mums looking after them, and then the rest will go out to feed. Doc also responds to sea lion sightings and puts out lots of signage and public information. This is another one of the critters I worked with. It is the yellow-eyed penguin, or the hoiho. So hoiho means noise shouter in Te Reo Māori, and it's a very good name. They're very noisy. You can hear them in the bush. They are the largest penguin in New Zealand and the 4th largest penguin in the world. Despite this, they only weigh about 6 to 8 kilos, so they're pretty little, and their populations are rapidly declining. They have individual nests, so there's no big breeding colonies. So a mum and a dad will go to a nice little spot in the bush. make a little nest, and have up to 2 eggs per year. Sometimes if eggs are not successful or they're not fertilized, or maybe something comes along and eats them, they can actually take one of the eggs from one penguin pair and give it to another. So they've done this for penguins that have just been released back into the wild, and also ones that maybe their eggs have failed. So the threats to the penguins, a lot like the sea lions, they are preyed on by stoats, cats, dogs, and ferrets. And in the ocean, they're prey for a number of really big animals. Like the sea lions, there's overfishing of their prey and a loss of habitat space. So pretty similar. So just like the sea lions, Dock and Yellow-eyed Penguin Trust do a lot of different work. They monitor the nesting sites and look for nests. They microchip and band the chicks, scan microchips on adults, monitor the molting and fledging of chicks. So that means when they lose feathers and they can't go swimming, and also responding to injured penguin sightings. So what was my ambassadorship? So I got to do a few pretty cool activities with the sea lions. These included monitoring a sea lion pup that was born at a golf course, searching for pups in the bush by following sea lion tracks, responding to sea lion sightings near busy roads, and I got to help tag 2 sea lion pups. So these are 2 of the pups that I met. We have got Hiriwa's pup, Hiriwa. So this pup here was born at a golf course. So it was near the 13th hole, busy golf course, lots of people, big walking track, as well as a lookout. So what did they do? They closed off the road. It's the first time a road has been closed for a sea lion, meaning that people couldn't take their cars up, and it minimized the amount of people disturbing them. There's also lots of signage around and dock staff sitting there, making sure that nobody was taking their dogs. Luckily, this pup has been moved to a crate and is doing really well. This one here, I like to call the flax nest pup. So we got to help tag this pup. In this photo, it's about a week old and looks just like a little puppy. And just like a puppy, they are super clumsy, super fun and playful. So we only tag the pups when the mums aren't around, otherwise they get a bit grumpy. So this here is the flax nest pup. Hopefully you guys on Zoom can see it. So this is just after we tagged it. You can see that even though we've just done a pretty painful procedure, we've put some flipper tags in and put a microchip in its bum, it's still pretty playful. It doesn't mind that we're there, and it's not too bothered either. So you might be able to see on its flipper, it has a little pink tag. So this goes through the muscle and has 2 layers of plastic on it, each with a unique code. Now this flipper tag will stay on as the pup grows up into an adult and allows us to monitor their sightings. If it's a female, we can also track who their babies are. So it gets one of these in both of its front flippers, as well as a little microchip in its bum, so just above the hip bones. And that microchip is just like on a cat or a dog, and they can scan it, find out exactly who it is, which is really important for monitoring sightings, but also if they're mums having pups, or if they're males and where they're hanging out. This here is Hiriwa and her pup. So this is just at the golf course. This is about 30 meters away from the golf hole with the little flag. And you can see they're not really bothered by us. We were a nice, respectful 30 meters away. And you can see mum's just hanging out there and her pup's having a bit of fun. So that pup would have been about 2 or 3 weeks old when this footage was taken. And you can see he's just hanging out. He's a bit bigger than that last pup that we saw. And they're just hanging out. So when this footage was taken, we were going to go and tag him, but mum was there. And mum weighs 250 kilos. And if we tried to grab her pup, she would probably give us a nice bite. But they came back the next day while mum was getting fish and tagged him. So he's doing really well. You might wonder, where are the dads? There is no paternal care with these sea lions. So that basically means it is mum's responsibility to look after the pup, feed the pup, and make sure it grows into a big, beautiful sea lion. Here you can see one of the females that we saw. Again, super relaxed and chill. They're not going to hurt anybody, but it is really important to give them plenty of space. In this footage, you can see that it looks like the sea lion is crying. It's got some little tear patches on its face. So this is how sea lions actually get rid of salt. So seabirds do it as well. When you're in the ocean and you're drinking seawater and you're eating fish, you get a lot of salt in your body. Best way to get rid of it is little tears. So it's not a sad sea lion, it's just one that's had a good drink. Males versus females. It's very obvious which one is which. The males are darker brown in coloration and look a bit fuzzy. And they're also quite a bit larger than the females, which are this beautiful light brown color. So there's not much monitoring of the males. They're pretty happy. It's more the females we want to look after because they're the ones that are going to have the babies. So they won't go and help mum feed the pups. In fact, they will actually try to find the females as soon as they've had their babies because that is the perfect time to mate. Basically, as soon as the pups are born, the mums are ready to get pregnant again. So the males will try and find the females, and if there's a pup around, it might get squished. So the females will hide their pups up in the bush and try to stay away from the feisty males. What's a good example of this? Well, when we were in Dunedin, there was a lovelorn sea lion led astray. So we actually went to go check this out. So we were part of this article, and here is Dock Ranger Jim Fife, my buddy Giverny, and a track. So this is what a sea lion track kind of looks like, big patches of flattened grass. But the male sea lion, he followed these tracks, thinking there was a nice pretty sea lion at the end, but it was actually quad bike tracks. So he followed the tracks all the way from the beach onto a busy road, hung out looking for the female, and unfortunately, the public had to come and push him away. So this is one of the impacts that sea lions face, not just human disturbance, but also being led astray by the quad bike tracks. So that kind of summarizes most of my sea lion work. So we'll get into the hoiho. So a lot of my work involved monitoring the nests to check on fledglings, but also the parents. So the parents will come and feed their chicks. If they've got 2 chicks, it's a lot of food to feed out. So you have to get plenty of food at sea. And if one of the parents disappears or decides not to look after the babies, it means that there will be one parent who has to work extra hard. We also transported underweight and injured penguins to the Wildlife Hospital and the Rehab Center. And we also took to the bush for early molters. So early molters are adult penguins that are coming in to get rid of that summer coat of feathers and get their beautiful winter feathers. So if you're not too clued up on penguins, they have 2 layers of feathers, a fluffy down, which is kind of like a thermal, and waterproof feathers on top, which are a bit like a snow jacket. So when they're getting rid of that coat, they have to stay out of the water, otherwise they will get very cold. We also helped clean up some poo at the penguin place and observe their daily husbandry needs. So this is a chick and this is an adult. They are the hoiho, so they've got that yellow band on top of their head and bright yellow eyes. So when they're a baby, it's a bit paler, paler eyes, band and beak. And sometimes they have this baby fluff as well. So when hoihos are babies, they are brown and fluffy, and they're not waterproof. So it's kind of like having a big, thick, woolly jumper on. This is an adult here. It's got its nice yellow band and bright yellow eyes. So we'll see the adults as well, helping to look after the babies, or maybe coming in to have a bit of a rest. Here you can see them in the dark. So this is in one of the nesting sites. We have got a beautiful brown fluffy baby here and a very cautious adult here. So this was the dad. He wasn't too happy about us coming in to look at the chick, but we had to look at the chick to make sure it was doing okay. So this chick here, it got a weight check to make sure it was growing nicely. We microchipped the adult to find out if it was the mum or the dad. And after that chick got a bit bigger, so a few weeks later, we came back and tagged it. This is a baby when it's a bit fluffier. So this one here is called the guard phase, so the parent is guarding the chick. But once they're a bit bigger, they're able to fend for themselves. The parents will leave them alone. So this is called the post-guard phase. You can see this baby here. It's got its adult feathers coming in, so that white belly, black back, but it also has the brown fluffiness. So over time, that will go away. The chick will become waterproof and can go out to sea to become a grown-up penguin. How do you find a nest? Well, you follow the poo. This is what a lot of our field sites looks like, crawling on your hands and knees through the bush and often through poo. Luckily for us, the dock rangers had done most of the searching, so we just had to go to the known nest sites and check out the babies. So flipper bands and microchips. So this little green thing here is a microchip wand. It is waved over the neck of a penguin around here to check for the microchip. So this is just like a cat or a dog. If you find a dog on the street, take it to the vet. They will wave around a microchip reader to figure out whose it is. This is the exact same thing for a penguin, except they're not owned by anybody. So when we're going into the bush, we're recording which penguins we're seeing. And if it's a baby, we'll give them their microchip as well. So that just goes into the neck around here and will stay for the rest of their lives. Now, some of the penguins, those at a site called Boulder Beach, got one of these flipper tags. So it's a bit different to the sea lion one, which is a puncture with 2 tags sticking off it. This is a band. So the reason why they're different is the sea lions are pups and they're going to grow a lot, whereas these baby penguins are fully grown. So if we were to tag it, tag a sea lion pup with one of these, they would probably not do too well. So this here has a little code on it, and the rangers can tell who is who from that code and also from the microchip. Other work that we did, we set up some trail cameras. So these just sit down and take photos when there is movement. Unfortunately, if it's a windy day, it also takes pictures of trees and flax bushes. But what it can show the rangers is who's coming in. Are both parents coming in or just one parent? Is there enough food for the babies? And what we actually found with one penguin is only one adult was coming in and both chicks were losing weight. What did we do? We took the chicks to the rehab center and we actually found that the parent coming in had a massive wound on its leg. It had been bitten by an animal, probably a barracuda, and actually couldn't swim that well and couldn't walk that well. So we found that parent and took it to the hospital as well. If it wasn't for the trail camera, we wouldn't have known that the adult was injured and we wouldn't have known to search for it. So because of this bad boy here, we saved 3 little penguins' lives, which is pretty cool. This one here is a pot of gross pond water that actually has mosquitoes in it. So just like in some parts of the human world, there is malaria, there's also penguin malaria. So it's called avian malaria and it is carried by mosquitoes. So what we do is we take some mosquito samples, the little wiggly larvae inside ponds, send them off to a lab, but before we do that, we put some poison in the water. It is an animal-friendly one, it only targets mosquitoes, and it basically means if there was malaria, it's okay, we've got rid of it. It only lasts for a few days, but it's plenty of time to wipe out those pesky mosquitoes. Some of the highlights, these are some juvenile penguins at Penguin Place. So you can see they haven't quite got that full yellow band. And here they are preening each other, which can be used to as flirting. But in these babies, it's probably just a way for them to show their affection and socially bond. This here is a chick that we saw in the Catlins. So this nest here was right on the coastline. So it really shows that hoihos can nest on the beach or on the rocks, as well as deep in the bush. So this one here was hiding from us, but it was looking through its little hole. And you might actually see at some stage its little sibling runs past. So there were 2 penguins at this nest, a boy and a girl. Oh, there he goes. And they weren't too keen to have their weight checks. But it's okay, they looked nice and pudgy, so we left them alone. But that was one of my favorite nest sites. It was also nice and close to the shore, so we didn't have to hike for too long. Some of the other critters I met was a whole load of kekino, or New Zealand fur seals. These 2 here are babies. So just like the sea lions, it is their breeding season as well over the summer. They're not monitored by DOC as part of the program I was in, and that's because there are lots of these and not many sea lions. I also saw a leopard seal. So leopard seals are deemed resident in New Zealand, but there's not many of them. Up here in Auckland, we have Ufa, who is a resident leopard seal, but they usually come onto the beach just for a few hours, have a bit of a rest, and then go back to sea to feed. We don't know too much about these guys, so it's awesome to get to come and see this one having a sleep on the beach. We also went to the Royal Albatross Center in Dunedin, and this here is the Royal Cam chick. So it was only a few weeks old when this photo was taken. He's doing a little squawk there. So this is a Northern Royal Albatross. They are the only breeding colony on mainland in the world, which is pretty cool. So this here is one of the parents. They look after the little fluffy baby, while the other one goes out to sea. So the best time to see an albatross is on a windy day, because that's how they fly. They don't flap their wings, they just soar through the air, which was really cool to see. My advice to applicants, get a great reference early. It makes it much less stressful before it's due. Also go out and explore your backyard. If you're a fish person, go for a swim at the beach, maybe get a scuba license or go snorkeling. Or if you're a terrestrial person, go out hiking, join a local tramping club, get some experience. The Blake Ambassadorship was made after Sir Peter Blake, who is one of the greatest explorers and sailors that New Zealand's had. Volunteer in your community. It's great to go out and join a local beach cleanup and put that passion of yours to action. And something good to figure out is, what is your why? So why do you do what you do? And show Blake your passion. Maybe you're passionate about art or design, volunteering in your community, and maybe you just like fish. So before I wrap up, this is a snares-crested penguin. So we weren't working with them on the Yellow-eyed Penguin Trust trip. So this one washed up really, really skinny on a beach, but it just really great, just really shows how fluffy they are and how cute they are. So I was pretty lucky to get to go on my trip, meet some baby penguins, adult penguins, and plenty of sea lions as well. Thank you for listening. I'm going to leave you with this little video of some kekano pups playing in a little rock pool. Thank you.
Thank you, Wednesday. We're going to open the floor again for some questions, and just remind everyone on Zoom to please type your questions on the chat box. I have a question for you, Wednesday. Can you tell us a little bit about the parental care? Does the father cares for the yellow-eyed penguin, or what's the mother contribution to the parental caring?
Yeah, so when they're in that guard phase, I'll quickly go back. So during this time, one penguin will always spend time with the baby. So if the dad is on the nest, the mum will go out fishing, get a big belly of food, come back, and then the dad will have a turn to go out. So it's not like the Empress, where the dads will look after the babies. They will take turns and share their effort. This is pretty good because it means that if one is doing a lousy job, the other can pick up the work. So that was pretty cool to see.
Yep, we have questions here. I'll repeat the question out loud for everyone listening. Janie Ann?
At the start, when you said sometimes they take, if they have 2 eggs and they're like taking eggs, like if they have 2 eggs, is there less chance of like them both surviving? Like will often only one survive?
Okay, the question is, if there are 2 eggs, if they lay 2 eggs, are there less chances of survival for the eggs?
Not necessarily. If they've got 2 penguins, 2 adults that are going out and getting plenty of food, both chicks can definitely grow into fat, healthy babies. If there's not quite enough food, then they will preferentially feed the bigger, bossier penguin chick over the weaker, shyer one, so they'll kind of favor it. But luckily with the dock rangers, if they see that one is struggling, they'll actually give it to either the rehab center or another penguin pair. They have had penguins in the past that have lost both eggs, so if they get a surrogate, they're going to feed it really well. The nest is super well monitored. If we know that a nest is there, then they'll do the best that they can to make sure both survive.
We have another question here.
Yeah, I think at the beginning of your presentation, you mentioned that you worked as a penguin keeper or assistant at the Kelly Tartans, was it? I was wondering what type of experience did you have to work there, or how did you manage to get that, get to work there at all?
Okay, the question is regarding Wednesday experience at the aquarium, and how did she get, or what kind of experience she needed to have to get that job as an assistant aquarist.
Awesome. For those who don't know, an aquarist is an aquatic fish keeper. So my job at the aquarium involves scuba diving in the tanks, feeding sharks, stingrays, endangered sea turtles, and sometimes helping out with the penguin department. So I was pretty lucky. I started working there in my second year of my marine biology undergrad degree. So I was studying here at Auckland Uni. The experience that I had, I did quite a bit of scuba diving. So for the role, they do require you to have a rescue certification, so PADI or equivalent. So it's really good to get some scuba experience if you're keen to work with critters under the water. Even if you're into mussels or mud, lots of scuba diving to be done there. In terms of special experience I had, I just really like fish and really like marine animals. And I also had experience keeping fish in the past, just little fish tanks at home.
Okay, more questions from the chat. It says, what made you decide what you wanted to work on the yellow-eyed penguin and the sea lions? Did Blake suggest these animals? If you chose them, what was so special about them?
So this is the first year they've run the hoiho and rāpoka, or New Zealand sea lion, ambassadorship. So they did do a hoiho one in the past, but this is the first year they've done it. So they did pick the critters for us. But I'm pretty happy because I would have loved to have worked on them anyway. So in Dunedin, their main projects are the hoiho, the New Zealand sea lion, as well as the albatross. So I guess they picked it in terms of what best aligned with the season we were going. If we went in the middle of winter, all the penguins would be at sea and all the sea lions would be in the ocean as well. So it's kind of just what worked best for Doc and Dunedin. But I loved working with them anyway.
One more question from the chat. It says, where do the hoiho nest? Is it just on the Otago Peninsula, or can they be found all around New Zealand?
So this is a pretty good question. So they actually were originally only found in the subantarctics. And then a couple hundred years ago, they started moving up to the south of New Zealand. So they are found all the way up to almost Christchurch, but this is kind of the limit of their range. In the subantarctics, they don't live on ice, they live in shrubs as well. So they are definitely a land penguin. They're not an ice penguin. But they can be found from the Catlins, so kind of on the western side of the bottom of the South Island, all the way up to Christchurch. But most of the nests are found either in the Catlins or on that Otago Peninsula near Dunedin.
And I have one more question. It's about the albatross. Is that the albatross place where you can actually watch them in camera, in the live camera? Yes, can you tell us a little bit about it?
Yeah, so the Albatross Center was actually one of the things that we did in our free time. So we were pretty lucky. We had weekends off, so we got to go on little adventures. So these photos here were actually just taken from the viewing platform at the Albatross Center. So this wasn't a special thing organized by Doc. That is what you can see if you go down to Otago. And this little chick here is the Royal Cam chick that is on the Facebook page and that Doc shares quite a bit of information about. It is now probably up to here on the adult, so it's definitely more than doubled in size, and it's actually being left alone now. So just like the hoiho, it's in the post-guard phase. So both parents can go out and get food while the little fluffy baby just hangs out in this nest here.
Awesome. Thank you very much. Wednesday, do we have any more questions for her? If we don't, can we all put our hands together in thanking Wednesday for her talk? Thank you, everybody who's attended today. We want to thank Amy, Janie Ann, and Wednesday, of course, for their amazing talks today. And keep an eye for the Blake Ambassador webpage. There's more information if you feel inspired to apply for next time around. And for everyone that's here, we invite you to stay. We're going to go and get some pizza for all of us. And thank you very much. Leave any comments on the chat box if you like, and we will be announcing any more things coming up for the Marine Science Society. And thank you for joining us. So thank you, everyone.